well, thank you all so much for joining today. I'm Megan, and thank you to OpenBox Science for allowing me to share my work and all the organizers. So I'm a postdoc at UC Berkeley. I worked in the lab of Kristen Scott, who's now retired, but I was when her lab was up and running, it was a taste neural circuit lab. So what that means is the general focus of the lab is understanding the feeding and taste circuit. So this could include anything from learning how taste information enters the brain, how it's integrated or processed and integrated with other systems, or then how it goes to share or inform feeding decisions with sort of monitoring the activity of different motor neurons to impact feeding. So I came from a background of reproductive biology, and I studied reproductive behaviors for a while. So when I joined the lab, I naturally established a project that was the marriage of those two worlds, so feeding and reproduction. So today I'm going to talk to you about changes in feeding that we see across reproductive states in females and in female fruit flies, Drosophila melanogaster. So initially, when I approached the field of research in reproduction, I did so from an evolutionary standpoint, from a framework, with the idea that the more we understand about female reproduction, the more we understand about the process of speciation. And that's very true. But beyond this, I think studying the change in a reproductive state allows us to understand things that govern state-related processes. So when we study a virgin to mated state, we can understand all this sort of latent infrastructure that allows for flexibility within an organism. So that's like genes, proteins, or neural circuits that can support this wide range of phenotypes. And we can understand the mechanisms, hopefully, that help to coordinate those state-compatible behaviors. So I think a really good example of that is food-related changes in the mated female. Okay, so the interest in mated-related changes in feeding is not new. We've known for about 20 years with radio-labeled food that mated females consume more than virgin females. And then since then, work from Carlos Ribeiro and Barry Dixon really asked what is in the food that females need more of. And they found that females, after mating, they establish a preference for yeast, which is the fly's main protein source. And similarly, they also develop a preference for salt. So these later two studies have really focused on the macronutrients that are vital to egg production. We know that egg production increases after mating. And then we know that when you restrict these two things, like protein and salt from the female, her egg production ability really reduces. Okay, so in addition to these sort of building blocks of egg production, we also know that females are going to need a little bit more energy. Eggs take a lot of energy to make and to lay. And there's a lot of other post-mating behaviors that will require additional energy. So in females, this increased activity results in an increased metabolic rate, driving the need for more calories. So this predicts that mated females will actually need more sugar than virgins. However, we also know that excess sugar in both humans and flies alike can really negatively impact the fly health. So this sugar intake should actually be tightly regulated. So although the ratio of sugar to protein has been investigated previously, the absolute amount of sugar intake in females, either virgin or mated, had not yet been investigated. So naturally, or very simply, when I joined the lab, I asked a very simple question: do mated females increase their sugar consumption? So to answer that, I used a automated feeding assay called the FLIC. It was originally made by the Pletcher Lab at the University of Michigan. And FLIC just stands for Fly Liquid Food Interaction Counter. So this is a top view of FLIC. The flies are placed, where's my screen? The flies are placed in this behavior arena. And then the liquid food is entered with into the food loading holes. And then with a sort of a canal, it presents the liquid food to each of the flies within the behavior arena. And this is really, really nice. It's automated, so it's unbiased. So we just get a result of how much the fly has eaten. And one other really good aspect of this is the fly is just freely moving around. So we can get a really good idea of their natural feeding behavior. So this is just a close-up to show you a little bit better how it works. So outlined in yellow is this liquid reserve. So this would be, in my case, sucrose water. And then around this is a metal ring. And so when we put the fly in, her feet are basically standing on the metal ring and her proboscis, or her mouth parts, are making contact with the sugar water. And you can think of this as like completing a circuit. So what we get is this, with time on our x-axis and this signal strength on our y-axis, we get this readout. And each of these sort of peaks are feeding events. And so we can get total number of feeding events. We can get average length of the feeding event. So it's total number and total feeding time. So what I did was I recorded the flies while drinking and compared it with signal strength. And I manipulated some of the sort of settings within R so that I could get a really good proxy of drinking time. So now when we have total amount time drinking, it is a really good proxy for the amount of sugar water consumed. Okay, so using this system, I then asked, does mating cause any increase or any changes in sucrose intake? So I have virgins here, and then I compared them with mated females, and I have total contact duration on my y-axis, which is going to be a proxy for amount of sugar consumed. And what I find is that there is a significant increase in how much sugar is consumed when I compare virgins to mated female. And so I wondered what was driving this change. So I wondered first if this was a drive dependent mechanism. So in this scenario, mating induces egg production, and then the egg production depletes energy stores. And then that would drive then the need for sugar consumption to restore homeostasis. So to test this, I then looked at females that don't produce eggs at all. So I looked at virgin eggless females and mated eggless females and asked, do they still increase sugar consumption? And what I found is that indeed they do. So it's not as strong as an impact, suggesting that there is something associated specifically with egg production, but I still saw a significant increase in egg production, in sugar consumption, suggesting that this increase in sucrose consumption is independent of egg production, meaning it's a drive independent. So there must be some sort of anticipatory response in flies that drives the increase in sugar consumption. So what exactly is that? Let's see. Okay. So I went into the sugar feeding literature to investigate on the involvement of any known hunger-dependent mechanisms that would drive consumption. So we know that hunger increases the sensitivity of taste sensory neurons to food, especially to sugar, when flies are hungry. So this increased sensitivity is thought to contribute to the increased consumption in hungry flies. So if mated females were consuming more based on this mechanism, I would expect the sugar sensory neurons to be more sensitive in mated females than in virgin females. So to test this, to test the sugar sensory sensitivity was underlying this increased sugar consumption. I monitored the taste-induced neural activity in sugar gustatory neurons in mated and virgin females. So I used two tools. Just used this GCamp. So I used a neural activity proxy of fluorescence and confocal microscopy. So GCamp is a neural activity indicator, and it translates neural activity to a change in fluorescence. So we can see this with a microscope and some lasers. So I drove this tool, GCamp, in sugar sensory neurons. And then I put the fly under the microscope. So I actually had to remove the top of the fly's head, of the cuticle, so I could actually image the brain in living, behaving flies. And then, as you can see here, I stimulated the proboscis. So this is the mouth part with some sugar water here shown in blue, with a glass capillary shown here as sort of a black box. Okay, and then I was specifically imaging a part of the fly brain. So here's just a very sort of simple version of a fly brain. And then I was imaging specifically this region called the subesophageal zone, or the SEZ. And this is really the taste processing center of the brain. So local neurons come in, and this is also where there's local circuits regulating feeding, and the motor neurons come out to feed. So I was imaging right here. And I was just asking, when the fly is, when the sugar is touching the proboscis, do I have, how much activity are those neurons showing? So here's an example of a virgin and mated female, and this is going to show you the change in fluorescence. So this is taking into account the background amount of fluorescence and then the increase in fluorescence that we see when the sugar is exposed to the proboscis. And when I quantify this information, so here I have time on my x-axis and change in fluorescence on my y, and this bar here is showing when the sugar water is actually in contact with the proboscis. And then these muted colors are individual traces, and then the bolded ones are the averages for mated, shown in gray, and virgin shown in gray, and mated shown in purple. And when we quantify this, we see that there is no difference in change of fluorescence, which is a proxy of neural activity. So both mated and virgin females show the same amount of neural activity and sensitivity to the sucrose. Okay, so this tells us that this sensitivity is not driving, this increased sensitivity is not driving the increased sucrose consumption in mated females. So the next mechanism that we then looked at is more in the world of fly reproduction. So there is a male-derived peptide called sex peptide. It's a 36 amino acid peptide that's made in the male reproductive tract and transferred to females during mating. And so once it's in the uterus, it really initiates this thing called a post-mating response in mated females. And that two hallmarks of that are an increased egg production and a decrease in sexual receptivity. And so we also know that these changes are initiated or maintained by female-specific neural circuitry. So what does that look like? So once sex peptide is here in the uterus, it can be sensed by these sensory neurons. So here's just a sort of an outline of a fly. We have her abdomen here with her reproductive tract. We have her thorax and her head, which then houses the brain and the ventral nerve cord, which make up the central nervous system of the fly. Okay, so here sex peptide is transferred to the uterus and is sensed by these sensory neurons called sex peptide sensory neurons. SPSNs for short, which then project up to the thorax, which then impinge on these second order neurons in the VNC called SAG, second order sex peptide abdominal ganglia. And then those then project up to the central brain and impinge on PC1. So this is a 3-tiered female-specific circuit that basically takes the signal that I have mated from the reproductive tract all the way up to the brain. And change in activity of this circuit influences egg laying and sexual receptivity. So I said, I wonder if this is also involved in the change of in feeding. Another really important point of this circuit, we know that it's active in a virgin female and then actually becomes silent in a mated female. So this is sort of a very simple neural circuit diagram that I'll continuously show throughout. These circles represent neurons, the arrows represent synapses. And so you have your sex peptide sensory neurons, SAG neurons, and PC1, and which are active, they're shown in green, active in a virgin female. And then upon mating and exposure to sex peptide, this circuit becomes silenced. So it's shown in pink. So it's silenced in a mated female. So I asked, maybe is this circuit associated or regulating post-mated female feeding? Okay, so this is just to remind you here of the circuit, active in a virgin female, silent in a mated female. Okay, so I decided to test this circuit one population at a time. So I started with the sensory neurons, sex peptide sensory neurons, and I was able to access these neurons with an intersectional method, so a sort of a combination of genetic drivers. So I was only accessing these neurons and no other neurons within the central or peripheral nervous system. Okay, so this is a very blurry picture, but here is actually the female uterus. And then up here are the cell bodies of these 6 to 8 sex peptide sensory neurons here, showing you I was able to genetically access these neurons. And then the goal was to basically make a pseudo-mated female. So I have the circuit here. So in an otherwise virgin female, so this circuit should be active. However, I'm artificially silencing this circuit with a tool, with a constitutive silencer. So I'm silencing these sex peptide sensory neurons. And then I'm asking how much sucrose do these females consume. So here on my x-axis, I just have the genotype, and again, my total contact duration, which is a proxy of consumption. And in gray, I have my genetic controls. So, and then in orange, I have the females that have these neurons artificially silenced. So these are all 3 groups are virgin females, but the ones with the neurons artificially silenced are sort of like pseudo-mated female. And what I find is that just silencing this population of neurons actually increased sucrose consumption, recapitulating the mated state in an otherwise virgin female. So this was very exciting. This suggested that indeed this circuit is involved in the post-mating increase sucrose consumption. So then I went on to test the rest of the circuit, SAG and PC1. Okay, so for the SAG neurons, instead of using this sort of complicated intersectional method, I was able to generate a different driver line called a GAL4 or a split GAL4 driver line, which allowed me to, in a simpler way, access just these neurons. So it's the same goal with a slightly different tool. And then the goal was then again, in an otherwise virgin female, artificially silence just this population of neurons and test changes in sucrose consumption. Because I was able to use a different tool, in this time I was able to use what's called a acute silencer. So instead of constitutively silencing these neurons, so instead of always silencing the neurons, I was able to only silence them at exactly the point that I was testing. To do that, I was using optogenetics. So I was using light to silence these neurons. And because of that, I actually had to create an LED light to go on top of the flick. So I can expose these flies to light and silence them only while the flies were in flick. And then also because of that, I was increased the assay time from just a 20-minute assay to a 4-hour assay. So I could look at changes over time. And what I found, so this is a slightly different graph. So I have time on my x-axis, and again, a change in feeding duration on my y-axis. Okay, so again, in gray, I'm showing you the controls. And then in this sort of mustard color, I have the females that were expressing this optogenetic tool just in the SAG neurons. These flies are also just showing this data set, this graph is also just showing a change in contact duration, so change over time, because each line is actually within genotype. I was exposing some flies to light and some flies to not light, and so this is the difference. So this is the increase in consumption that I saw in the light condition minus the ones in the dark. So this graph is not only showing me genetic controls, but it's also showing me light controls. And what I do find from it is that compared to controls, the flies that had their neurons silenced showed an increase in consumption over time, suggesting that silencing the SAG was actually driving sucrose consumption. That was very exciting. And in much the same way, I was able to test PC1. A split line was already made by the Barry Dixon's lab for PC1. So I was just, again, silenced this neuron in an otherwise virgin female. And again, I saw an increase in sucrose consumption, suggesting, so putting that all together, it suggests that indeed, this increase in sucrose consumption that I'm seeing in mated females is really being initiated by the activity of this circuit. Okay, great. So next, naturally, in a circuit lab, you always ask what's downstream. So ideally, I wanted to connect this circuit to some sort of feeding regulation circuit, either a circuit that goes back to the SEZ, so that region of the brain that I was originally imaging for, that has the taste sensitivity experiments, or maybe it connected to some sort of neuroendocrine circuit, because we know that hormones can impact both a hunger state and feeding behavior. Okay, so what's downstream? Recently, there's been this incredible data set that was generated by Genelia, where they took a female fly brain, sliced it, imaged it with electron microscopy, and then put it all together to create this sort of digital representation of the fly brain and create this connectome. And so I was able to then use that tool to ask what's downstream. So this is just an example of what this data set would look like. This is using Flywire, but here is just an EM slice, and you can see all these different colors. They're actually different neurons within this data set. And then to the right, we can see a 3D neural reconstruction of its actual morphology with the cell bodies here. Another part of this data set is that you can visualize synapses, so you can get not just what the neuron looks like and where it is, but who it's getting information from and who it's giving information to. So I was able to query this data set and ask, what is downstream of this mating status circuit? Okay, so just to remind you of the circuit, and then in the whole brain here, we could visualize both, we could find both SAG and PC1. So with help from the Barry Dixon's lab, we were able to identify both these neurons within this data set and then query it and say, what's downstream? Okay, so here is sort of another simple circuit of SAG and PC1. So again, my neurons are shown as circles, and synapses here are shown as arrows, and the number beside it is the number of synapses between those two neurons. So we could find SAG and PC1 and all of its potential downstream synaptic partners. So one cell type popped out to us. It's a single cell type that has 3 neurons. So all these neurons look very similar to each other, and we just sort of labeled them A, B, and C. And then this is a sort of a more simplified version of the circuit. So this is my original circuit, and then we know that this cell type is downstream of both PC1 and SAG. Additionally, from the literature, we know that SAG and PC1 are cholinergic, meaning when they're active, they're going to activate or excite anything that's downstream of them. So we then hypothesized that these neurons would actually, this new cell type would also be active in a virgin female and silenced in a mated female. So what do these neurons actually look like? So here are images from this data set. So it's a digital sort of reconstruction of A and B of this cell type. So I said that there was 3, A, B, and C. I'm just going to focus on A and B for the rest of the talk. They're the most strongly connected downstream neurons. Okay, so what do they look like? Well, we went into the literature and we found this neuron called PCD2. We think that they have the same projection pattern, they have the same number of cell bodies, so we think that they're indeed these neurons. So that was very cool. They have names, so then we named this one PCD2A and PCD2B. And then I set out to make a split Gal4 line, so a genetic driver where I could access these neurons and only these neurons in the whole entire nervous system. So I was able to do that. So that's very exciting. And now I can test, are these neurons actually downstream of PC1? And do these neurons impact feeding behavior in the fly? So I was able to use some of the an optogenetic tool and GCamp to measure changes in activity, just like the same tools that I've used before. And these experiments were actually done by Gabriella Stern, who's now at University of Rochester, and who's in the chat today. So thank you, Gabriella. So she was able to activate these PC1s using optogenetics and light, and then measured changes in activity in these potential downstream neurons. It's called functional imaging experiments. And what we find is we have time on our x-axis and change in fluorescence, so activity of this downstream PCD2 neuron on our y. And we see that during these gray shaded times is when the light was activating PC1, we definitely see an increase in activity of these potentially downstream PCD2A neurons. And when we quantify that, we see a significant increase. And we did the same thing for PCD2B, saw a very similar activity pattern. And so we can say two things from these experiments. One, that they are downstream, so they're functionally connected to PC1, and two, that they are connected in the same way in terms of activity in PC1 equals activity in PCD2. So just like we hypothesized. So then we asked, do changes in the activity of these neurons impact changes in feeding behavior? And so I again put them into Flick with my optogenetic setup where I'm taking a virgin female and artificially silencing just these PCD2 neurons and looking for changes in feeding. And again, I saw a significant increase in feeding when these neurons were artificially silenced in an otherwise virgin female. So this is fantastic. So we found potentially the output of PC1 that's regulating post-mating feeding in females. But these neurons weren't known to impact feeding. So I wanted to keep the circuit going even further to see if I could reach the SEZ or this neuroendocrine system to really understand how mating impinges on feeding regulation. So I went back to this sort of like connectome, so this data set that represented, so this digital representation of fly brain, and I asked, what's downstream of this cell type, of this PCD2 cell type? So the biggest sort of recipient of information from this cell type is this collection of neurons. It's 31 neurons per hemisphere, so 62 total. And we went back into the literature and we said, what are these neurons? A population of neurons based on anatomical similarity were these sort of median bundled neurons that express this insulin receptor, LGR3. So that was really exciting because these, for a few reasons, these neurons are sexually dimorphic. So anything that's sexually dimorphic often means that it is influencing some sort of female-specific behavior. And LGR3 being a receptor for an insulin protein or insulin peptide, there was clear ties already to feeding regulation. So LGR3 is a receptor for DILP8, Drosophila insulin-like protein 8. And when we mutate, when previous studies have mutated either DILP8 or LGR3, we see an increase in feeding, suggesting that DILP8 and LGR3 are sort of part of a satiety system, or that DILP8 is a satiety factor that regulates feeding. So this was very exciting. So we wanted to say, we wanted to ask, are these neurons functionally connected? And can I influence feeding when I manipulate their activity? So again, Gabriella did some functional imaging connectivity experiments, changing the activity of PCD2A, so exciting PCD2A, and then looking for changes in activity in LGR3. And this time what we found is that when PCD2A were activated, we actually got a decrease in fluorescence, suggesting that that PCD2A was inhibiting the activity of LGR3. And we saw the same thing for PCD2B, but slightly less. So this would give us a general circuit pattern of in virgins, this circuit is activated, and then we have this active inhibition of LGR3. But then in mated females, this circuit silence, and that alleviates the inhibition of LGR3. So that in a mated female, LGR3, these median bundle neurons are active. So I did a similar optogenetic experiment where I'm activating these neurons in an otherwise virgin female. And again, I saw an increase in sucrose consumption. So that was really exciting. So I was able to go from mating circuit all the way to some sort of hunger center in the fly brain. So just a quick summary. What I found in general was that mated females consume more sucrose than virgin females. This change is independent of egg production and is regulated by the mating status circuit. I identified this output of the mating status circuit, the PCD2 cells, which impinge onto these median bundle LGR3 cells, which is the DILP8 receptor. And I was able to then, what this sort of bridge represents is the ability for coordination of mating state and sugar or caloric intake. And so that these females can basically match their nutritional intake to their energetic requirements in order to maintain sucrose or caloric homeostasis, as well as improve reproductive success. Okay, so this is the Scott Lab that no longer exists, but big thank you to Kristen Scott, wherever she is in the world, and huge thanks to Gabriella Stern, who was an incredible help during this project, and then our old lab. And thank you all for listening.